Okay, today we're going to look at the brain. And we're going to talk about the human brain, but we'll use the sheep brain as a comparison because it's what we'll have available to dissect. So let's talk first about the parts of the adult brain. These are the four adult brain structures. They consist of the cerebrum, which actually has two cerebral hemispheres, the diencephalon, made up of the thalamus, hypothalamus, and epithalamus. And the brainstem has three structures as well, the midbrain, pons, and medulla oblongata. And then the structure, sometimes called the little brain, the cerebellum, is behind the cerebrum, and it consists of inner white matter that kind of looks like a branching tree and is actually known as the arbor vitae, which means tree of life. And then there is the outer gray matter, or the cerebellar cortex. So those are just the basic parts of the brain, and we're going to look at them in more detail. So here are those four main regions, so we can visualize them. Here we're seeing just the left side of the brain, so we can only see the left cerebral hemisphere. In this dark purple is the diencephalon, so we're not seeing the parts like the thalamus and the hypothalamus and the epithalamus sectioned out just yet. Then in red, behind the cerebrum, is the cerebellum, and you can see its size is much bigger than any of the other structures except for the cerebral hemispheres. It's the largest and topmost part. The cerebellum sits just behind what we actually call the occipital lobe, and the cerebellum is going to be also behind the brainstem. At least it is in the human brain. A sheep brain actually has a horizontal brainstem, whereas ours here is vertical. And so these structures in green, the midbrain, pons, and medulla, those are the brainstem. The medulla oblongata is actually continuous with the spinal cord. And even though the spinal cord is very important, it's a part of the central nervous system, just like the brain. It's not actually a part of the brain itself. It's just an adjacent or associated structure. So let's look at those four main regions and talk about some general functions. The cerebrum, or two cerebral hemispheres, that's what does all of our thinking, especially the outer gray matter in the cerebrum. The diencephalon consists of structures that are the relay station for incoming sensory information, the location for a lot of homeostatic mechanisms, the main control for the autonomic nervous system and the endocrine system, your thirst and hunger center. All of those functions, they fit the hypothalamus. And the epithalamus contains, at least mostly, the pineal gland. And this is a gland that secretes melatonin, and that helps regulate your sleep and wake cycles. We're looking at our brainstem. We have the midbrain, which have a lot of interesting structures, and they're pretty fun to say, like cerebral peduncles or corpora quadrimina. The corpora quadrimina is actually made up of two top bumps and two small bumps that are below the top bumps. And they have names like superior colliculi and inferior colliculi and contain a lot of visual and auditory reflex centers. Below the midbrain is the pons. That's really just a conduction tract to communicate between the midbrain and the medulla. And the medulla has a lot of respiratory, cardiovascular, and other important autonomic centers that help regulate things like heartbeat, heart rate, respiratory rate, and depth. So any damage to this medulla can be very fatal. And the cerebellum again has the arbor vitae, which allows for communication across that entire cerebellum, and it's going to function primarily subconsciously. And it creates a visual muscle blueprint that it communicates to our cerebrum to help our body know how to move. We can also look at spaces that are within the brain. And these spaces within the brain are called ventricles. And these ventricles are filled with a fluid. This fluid is kind of like blood plasma, but it does differ in the amount of proteins and ions, but it is similar in consistency if we were looking at what it contains. This fluid in the brain, specifically in the ventricles, is called cerebrospinal fluid. And it's something we find not only in the brain, but around it. and it's going to reduce the weight of the brain by about 97% and allows it to float in its protective skull. It also is going to nourish the brain. And again, while this cerebrospinal fluid doesn't have all of the components of blood plasma, it has things like potassium and calcium ions, sodium and chloride, and hydrogen, and that's enough to nourish and allow for electrical potentials to be generated in the brain. Now, the ventricles of the brain are arranged in certain locations. You have two C-shaped ventricles in the two cerebral hemispheres. So let's look at those. On the left image, we have one C shape on the left, and then on the still left image, on the right hemisphere, we have another C shape. And they're going to be connected by a tiny hole called the interventricular foramen. You don't need to know that term, but I just wanted to point it out. So our first and second ventricle are called lateral ventricles, and they're C-shaped, and there is one in each hemisphere. The third ventricle is located closer to the diencephalon, where earlier we saw that purple structure. That's where we'd see the third ventricle. The opening for this is actually above the thalamus. And the third ventricle is connected to the fourth ventricle by a canal called the cerebral aqueduct. We can actually see the fourth ventricle is quite wide, and it's located at the base of the cerebellum. So we can see now that there are four ventricles in the human brain, two lateral that are C-shaped, the third and fourth ventricle. And actually coming off of the fourth ventricle is a central canal. And this central canal goes down through the spinal cord, and it's a tiny cavity within the spinal cord that also contains cerebrospinal fluid. Now let's look more closely at those parts of the brain. So for example, the cerebrum kind of looks like a wrinkled walnut. It's very convoluted or folded, and we have names for the folds and also the grooves that are between the folds. A gyrus is a ridge, and a sulcus is a groove between ridges. And the gyri and sulci of the cerebrum give it this convoluted shape. The reason our brain is so convoluted and folded is to allow for more brain matter to fit in a smaller space. Because our brain grew so quickly during the beginning of development and still had to fit within our skull, we saw these convolutions form. There's also a couple of really deep grooves, and deep grooves in the brain are actually called fissures. There is a longitudinal fissure that separates the left and right hemisphere, and there's also a transverse fissure that separates the cerebrum from the cerebellum. And the two cerebral hemispheres are actually connected, but only by a thin piece of tissue in between them. And the cerebrum is the collective name for the left and right hemisphere, whereas hemispheres generally talks about one or the other, not both, but we do have two cerebral hemispheres. Here is that piece of tissue that connects the two hemispheres. Now, it's known as a piece of white matter, and its name is corpus callosum. I know it doesn't necessarily look like white matter here, but it is lighter in appearance compared to the outer gray matter. And this is actually the only true physical connection between the left and right hemispheres. And when you're looking at the brain, we actually find that there's gray matter on the outside and white matter on the inside. So it makes sense that this internal structure is made up of white matter. And more on what white and gray matter is in a moment. Here's the basic arrangement of gray and white matter inside the brain. You see the darker gray matter on the outside and a little bit in certain kind of island locations on the inside. And then you see mostly a lighter material on the inside, and that's the white matter. This gives the brain three general regions: the outer cerebral cortex, the inner white matter, and these islands of gray matter, those are called the basal nuclei. We can actually see a side view of the corpus callosum here. Here's the only true physical connection between the hemispheres. What is meant by only true physical connection is that's the connection they use to communicate with each other. Now, what is gray versus white matter? Well, it's really just where do neurons and their parts accumulate. In the gray matter, we find mostly the cell bodies and sometimes dendrites. These are the receptive regions of a neuron. They're not the parts that are going to be carrying long electrical signals to other neurons. They may start or initiate the spark, but they're not the long processes. The long processes tend to be covered with myelin, which is a fatty substance that looks white under a microscope. And these long processes called axons appear in bundles, and all of this white myelin groups together, giving the entire area or tissue a lighter appearance. So gray and white matter is just really based on what's accumulating where. Areas without the fatty substance appear darker or like gray matter. Areas with the fatty substance appear lighter or like white matter. And we actually see that someone's cerebral cortex or main gray matter inside their brain doesn't even really develop until their 20s. Whereas the white matter is going to develop throughout the 20s. It actually peaks or develops most fully in middle age. This is when we see a lot of connective pathways in the brain really come to fruition. The main function of gray matter is to conduct and process and send information to different parts of the body, but only after integrating and deciding or making a decision. Whereas the white matter, it tends to be used to get sensory information to the brain or send motor information back to the body. So the thinking part of the brain is the gray matter, and the white matter is all of the conduction pathways, essentially the neuron highway. We can also look at the lobes of the brain. There's 5. Now what we can see here is just 4 of them. The frontal lobe in yellow, the parietal lobe in pink, the occipital lobe in purple, and the temporal lobe in green. You actually have 2 temporal lobes. There'd be another on the left cerebral hemisphere. There's also going to be a lobe that we can't see that's located under the green temporal lobe. That 5th lobe, it's known as the insula or insulary lobe. If we take a closer look at the brainstem, we can look at its 3 parts. In the human brain, we have the superior or topmost midbrain, which controls involuntary eye movement, and then reflexes related to both visual and auditory information. Then in the middle, just under the midbrain, would be the pons, and this relays messages from the cortex, that top cerebral part, to the cerebellum, but it's also responsible for things like facial sensitivity and plays a major role in rapid eye movement sleep, and that's when dreams occur. It's mainly a conduction pathway, but it does have some functions of its own. And then under or inferior to the pons is the medulla oblongata that regulates things like breathing and heart rate, digestion, sleeping. It even regulates muscle tone, types of movement. It initiates reflexes related to vomiting, swallowing, gagging, coughing, sneezing, and hiccuping. A lot of automatic responses we don't think about needing to control. It is actually going to be something related to the autonomic nervous system, which does stuff automatically or subconsciously for us. Let's look at the actual structures in a little bit of detail. We can see here in kind of this maroon pink the diencephalon. So in the human brain, the midbrain, pons, and medulla sit under that diencephalon and in front of the cerebellum. We take a closer look, we can actually see the opening into the 4th ventricle is behind the pons here in blue. A lot of the respiratory centers related to the medulla oblongata have some neighbors in the pons, but again, most of those automatic or autonomic functions belong to the medulla oblongata, which receives higher signaling from the hypothalamus. And when we're talking about the parts of the brain and the lobes and their functions, we can actually assign general functions to those lobes. A lot of what the frontal lobe does is allow us to think, store memories, and is responsible for our behavior. The hearing, learning, and emotional part of our brain tends to be near our temporal lobe. Language and touch and visual processing, balance coordination, all of that tends to be towards the back of the brain, with language and touch being primarily the function for the parietal lobe, visual processing happening at the green occipital lobe. So we don't really have eyes in the back of our head, but that's actually where we process visual information. And then the cerebellum is going to take care of balance and coordination, a part of that blueprint it creates that it then can communicate to the upper parts of the brain. And the brainstem is more for autonomic functions. A lot of what the medulla does are the general functions that are oftentimes mentioned for the brainstem: breathing, heart rate, and temperature regulation. Again, those are generalizations. We can be more specific when we start looking at parts of the brain. Like instead of saying thinking and memory and problem solving, for the cerebrum, we could say responsible for conscious thought processes, memory storage and processing, responsible for communication related to skeletal muscle contraction, and subconscious regulation of that, which means communication with the cerebellum is required. We look at the diencephalon. That thalamus is a relay station for sensory information. What that means is all information coming up the spinal cord and then coming up the brainstem will eventually hit that central structure in the diencephalon that is called the thalamus. The thalamus then makes decisions and sends out that information to the right lobe of the brain so it gets processed the way it's supposed to. So it would send visual information to the occipital lobe, but not to the temporal lobe or the frontal lobe because that would be the wrong location for its processing. The hypothalamus has a lot to do with homeostasis, which means regulating internal body functions, controlling things like thirst and hunger and emotions, wakefulness, even hormone production. It's actually considered the autonomic nervous system boss as well as the endocrine system boss. The hypothalamus does a lot for body regulation. We can also look at things like the pons, which is the relay for sensory information between what's above it, the midbrain, also known as the mesencephalon, and below it, the medulla oblongata. The mesencephalon processes visual and auditory reflex information. It's also going to be an area that maintains consciousness, that essentially is regulating whether or not we're really awake and alert, or tired and sleepy, or actually sleeping. And then the orange medulla oblongata is going to be responsible for relaying sensory information. It is a part of that brainstem. It's a conduit to get up to those higher centers, but it also has all those autonomic reflex centers related to both the cardiovascular and respiratory center. And then the cerebellum coordinates complex somatic motor patterns. That is another way to say it creates a blueprint for motor activity. So it creates the blueprint about how to ride a bike, for example. For example, it may tell your brain, the higher parts of your brain, to move your knee a certain way, contract this muscle, bend this muscle in this order. That's what is meant by a blueprint. It actually even adjusts this output as the information leaves the brain and goes to or down the spinal cord. So now let's look at the basic human brain. Now we're looking at the underside in this view. Some things we can see that we haven't looked at yet are things like the olfactory bulb, which is a structure at the end of the olfactory nerve, and that's related to smell. Our olfactory bulb's pretty small compared to the size of our brain. The sheep olfactory bulb is going to be much larger compared to its brain size. Another structure we can see is this X shape. That is the optic chiasma. The top parts of the X are actually the optic nerve, and they would go out to your eyeballs. And we see that the X shape here means that those optic nerves at one point will cross before that information is sent towards the back of your brain for processing. Some structures we can see that we've actually talked about, like the cerebellum, which takes up about 11% of the brain's mass, the medulla, the pons, and even these mammillary bodies we haven't looked at specifically, but they're just a part of the midbrain. Here's a side view of the human brain. We can see the convoluted and folded cerebrum. We can see the corpus callosum above the lateral ventricle, which is covered by a septum pellucidum, which is just a membrane cover. In the very center, something that's I would always recommend you try to find first is a roughly circular structure, and that is the thalamus. And if you can find the thalamus, under it is the hypothalamus, because hypo means below, and then a little bit back and up is the pineal gland, or the epithalamus. Also, if you can find the thalamus, instead of moving forward and down, move straight down, and you found the mesencephalon, or midbrain. Then move down further, and you found the pons, which is usually recognizable by a pretty noticeable bump at the base of the brainstem. And then under the pons is the medulla oblongata. We can also see here a cross section of the cerebellum. You can see the lighter tree or branch structure in the middle. That's the arbor vitae. At the base of the cerebellum is the opening to the fourth ventricle. We can also see structures like a gyrus and a sulcus. So these ridges are gyri and these grooves are sulci. This brain has been separated along what we consider the longitudinal fissure, but here would be the transverse fissure. Here we can see a sheep brain, so we'll make a sort of transition here so we can look at what our dissection may appear as. Some of the things we can see here are the meningeal coverings, which we haven't really talked about. These are protective coverings, and there's 3 of them. From the outside in, they are the dura mater, arachnoid mater, and pia mater. Other structures we can see here is that the meningeal layers are embedding themselves in the longitudinal fissure, the cerebrum here with its many lobes, cerebellum behind, and a little bit of the medulla oblongata as it continues and forms the spinal cord. So the meningeal layers are sometimes just called the meninges. Again, from the outside in, we have the dura, arachnoid, and pia mater, and they're going to sit under your skull, closer to your brain than your scalp. The meningeal layers protect the brain, but they also are all over the spinal cord. A lot of them have web-like structures. There's blood vessels within or between, and they're going to have cerebrospinal fluid all around them to act like a cushion for the brain. The tough outer layer, that's the dura mater. A slightly thinner layer is the arachnoid mater, and the most delicate layer is the pia mater. And it's actually going to go into or fold over the sulci and the gyri. Here is the underside of a sheep brain. So you can see a little bit smaller. And compared to the size of the brain, the olfactory bulbs are huge. The optic chiasma, also much bigger. The optic nerves, a little less severed. We can also see the pons, which means the midbrain should be somewhere in here, followed by the medulla oblongata, which is continuous with the spinal cord. Here's another structure to look at, and this is a cross-section of the sheep brain. So it also looks a little different than the cross-section of the human brain. We can see the brainstem down here is much more horizontal. We can see the cerebellum is quite large for the size of the cerebrum, and we can see a lot of different labeled structures. So let's point some of them out. The lighter branch structure in the cerebellum, that is the arbor vitae, made up of cerebellar white matter. 2, we could call that just the cerebellum, but it's also the cerebellar gray matter. It's the outer cortex. Down here, 11, that's the opening leading into the fourth ventricle, is this little slit at the base of the cerebellum. Next, we can move down and look at the brainstem. This area here, including 3, 4, and 10, that's all the midbrain. 3 is a bump called a superior colliculus, and 4 is a bump called the inferior colliculus. And that's where the visual and auditory reflex centers of the midbrain would be located. 24 is the pons, and the medulla would be just behind that. And we can see how there's a small bump here before it smooths out, and that would be the spinal cord at the end. Other structures we can easily see is this large central circular region. 25 is the thalamus. 5 is the pineal gland or epithalamus. Then we have 20 and 15. They are on top of and under an opening, which is the opening into a lateral C-shaped ventricle or one of the cerebral hemispheres. The corpus callosum is 22, but proceeds downward, and the fornix is 15. The mammillary body is 17, also a part of the midbrain, and then 18 is the hypothalamus, which is under 25, which was the thalamus. Then you have 23. That's a side view of the optic chiasma. And we're seeing just the side view. So if we were looking at the thalamus as a starting point, you could say thalamus, hypothalamus, optic chiasma if you're moving forward. And then you could say thalamus, hypothalamus, and then you could say mammillary body if you're moving backward. Now, not all of these structures will be as visible in your cross section. It depends on how well you do your sectioning. You want to separate the left and right sides of the brain using the longitudinal fissure as your guide. Here's another view. Here we see not numbers, but actual labels. Superior and inferior colliculus, cerebral aqueduct under it, pineal gland, fornix, corpus callosum. Those are above and below the opening to the lateral ventricle. But if your lateral ventricle is covered by a membrane, that's the septum pellucidum. The folded wrinkle shape, that's of the cerebrum. Then you have optic chiasma, hypothalamus, mammillary body, thalamus, cerebral peduncle, which is also part of the midbrain, just like the colliculi are, pons, medulla, and spinal cord. And it's going to take some time. I would recommend looking at these structures or pulling up a practice image on Google. You would just need to type in sheep brain dissection labeled. Now here, before you do your dissection, you could actually pull back the cerebellum and actually see the superior and inferior colliculi better before they're cut in half. The superior colliculi are a group of two big top bumps, and the inferior colliculi are a group of two smaller bottom bumps, and they sit just in front of the cerebellum. The superior and inferior colliculi are where you find the visual and auditory reflex centers of the brain, and they are a part of the midbrain. The collective name for all four bumps is the corpora quadrumana. And the epithalamus, specifically the part called the pineal gland, would sit just above them. We can also see some labeling for the cerebellum here, and there is a medial lobe that is going to connect the two lateral lobes, and that medial lobe with the transverse or side-to-side fibers is known as the vermis. And then one of our last topics will be looking at the cranial nerves. There are 12 cranial nerves. There is olfactory, optic, oculomotor, trigeminal, trochlear, abducens, facial, hypoglossal, vestibulocochlear, glossopharyngeal, vagus, and the cranial or accessory nerve. So these are our 12 cranial nerves. Now some of them tell you what they do, like olfaction is smell, optic is vision. When I see cranial accessory, I think of a woman putting an accessory on her shoulder, like a purse. And that's near your trapezius muscle that allows you to shrug your shoulders. And that's actually one of the functions for that cranial nerve. Anything that has the prefix or suffix glosso, something to do with the tongue. Pharynx, that has to do with the throat. The facial cranial nerve allows for facial expression. And the trigeminal That has to do with mastication or chewing, and it has an M right in the middle. So there's different things about the cranial nerves that as you learn what the terms mean, help you remember what they do. The vestibule in the ear is related to processing information about equilibrium or balance, and the cochlea is for hearing. So this nerve processes both equilibrium or balance information and hearing. And the vagus nerve you study a lot if you study the parasympathetic nervous system, because it's actually going to be able to send signals not only to your head and your neck, but down into your chest, to your heart, and to a lot of your abdominal organs. So the cranial nerves are very interesting, and again, you do not have to memorize them, but they are an interesting course of study. You can see here that they're listed in order from 1 to 12. You can learn the cranial nerves by name or by Roman numeral. For example, olfactory, optic, and oculomotor are Roman numeral 1, 2, and 3. And if you said cranial nerve 2, you would mean the optic nerve. The optic, oculomotor, trochlear, and abducens nerve, or cranial nerve 2, 3, 4, and 6, they all have something to do with the eye. Optic is for just vision, it's only sensory. Oculomotor can actually move 4 of the 6 muscles that move your eye. It also can change the shape of your lens and the diameter of your pupil. Trochlear and abducens both move 1 of the 6 muscles that move your eye. Trochlear moves the one called the superior oblique, and abducens the lateral rectus. Trigeminals for chewing, but also some sensory information that comes from the face. Facial is probably the easiest cranial nerve to remember because it allows for facial expression and also for stimulation of different glands, like that in your nasal cavity, your tear duct or lacrimal gland, and salivary glands. And it allows for anterior taste buds to work. Then we have the vestibulocochlear nerve, used to be known as the auditory nerve, and it is going to allow for equilibrium and hearing information. It's mainly sensory, just like the optic nerve. Olfactory is also going to be sensory and allows for smell. Glossopharyngeal is going to send information from the back part of your tongue and your throat that is motor and allow it to move. It allows for swallowing and speech, with some taste and general sensory information. And the vagus nerve goes down to your abdominal viscera. It innervates your heart and parts of your neck, but also it's related to motor information concerning swallowing and speech. Your spinal accessory nerve or accessory nerve has to do with innervation and just motor innervation to your sternocleidomastoid and your trapezius muscle. The sternocleidomastoid is a skinny neck muscle that allows it kind of to move side to side. And the trapezius muscle is a diamond-shaped back muscle that allows you to shrug your shoulders. And so movement of these muscles is controlled by cranial nerve 11. And then cranial nerve 12 has hypoglossal, so it's something about your tongue, and hypo means below. It actually has to do with movement of your tongue. A lot of muscles located below the main structure allow for that tongue movement related to speech and swallowing. So again, you don't have to memorize these, but they're very interesting courses of study and something that if you ever have a chance to pick a topic, maybe something you want to look at more. And that is it for our look at the brain. So it's very complicated and we took a very topical view, but it's something that's fun to learn about because it's something that dictates how we behave in our environment and how we respond to things throughout our lifetime. So get ready for a brain dissection this Friday, and we'll take a closer look at the brain.